So, I want to start off and start talking about Mendelian genetics, and I again start off with usually a little bit of a fun PowerPoint, kind of like this artistic version of Gregor Mendel, simply because we tend to think about him as being a really serious guy, and this is sort of a little bit of a throw-off. Better picture here of Gregor Mendel. He was born in the 1800s. He was a Czechoslovakian monk, known as the father of genetics. At this point, I just want to introduce some of his ideas and give you some definitions, and then we'll go through and do lots of problems in class and in laboratory. But let's go ahead and get started with this guy, Gregor Mendel. He was basically, when he started doing his work, the alternative theory to inheritance to what he came up with was known as blending. So if a tall plant and a short plant were crossed, you expected a middle-sized plant, and that does occur sometimes, but it doesn't explain everything. He wanted to do some experiments. He was doing his work in the 1850s. What was interesting is his work, it was about the same time as Charles Darwin's. Their work supported each other, however, they didn't know about each other, and neither one of them shared each other's work. So let's talk first about the plant that he worked with. He worked with a pea. A pea is an interesting plant because both the female and male structures, the female structures are produced in the carpel, which is the pod. The male structures, the pollen, which are produced in the stamen, are found within the same flower. And the flower actually has petals that cover over the male and female parts, so self-fertilization is the norm for the pea plants. And if you get a true breeding set of plants, it's relatively easy to do because of this self-fertilization. The neat thing with this is you can also take the male reproductive parts away as they're developing before they begin producing pollen, and then you can control fertilization. And that's one of the neat characteristics of the pea plant. Mendel identified 7 traits. He looked at wrinkled and smooth seeds, green and yellow seeds, wrinkled and smooth pods, green and yellow pods, purple and white flowers, different flower types, and also tall and short plants. He isolated true breeding plants, plants that only showed one form of a trait, and he allowed those to self-fertilize, and he always got the same type of plants. And then he started doing crosses one between the next. So what he did is he deliberately transferred pollen from one to another and prevented the plants from self-fertilization. He allowed seeds to form. Then he grew on several generations of plants. So a couple quick things we need to introduce. We need to introduce the letters that are on the chart on the far right. So you see the letter P. Because we're talking about several generations and it's hard to sort of figure out who the parents are in a multi-generation chart, he uses the letter P for the parental generation, and F1 is first filial. First filial means first set of kids. Filial is set of kids. The F1 he allowed to cross and have a second set of kids, so you'll see that there's F2s as well. So that would be, in human terms, the P would be grandparents, the F1 would be parents, and the F2 would be the kids. But again, we use the letters instead. In this case, you can see he crossed a round and wrinkled seed, and 100% of the F1 generation was round. Again, that didn't follow what he expected based on the blending theory. So he determined that all the F1, all the parent generation, had heterozygous or had hybrid offspring that had characteristics from both parents. The one that he observed that the trait that masks the presence of another trait is referred to as the dominant trait. So a dominant trait masks the presence of another allele or another trait, and a trait that can be masked is considered a recessive trait. So the dominant trait is what you see, the recessive trait is present, but it's not shown. Okay, so when he allowed the F1 from that first cross to self-fertilize, he counted all the seeds, and with 7,324 peas, he found 3/4 of them were round and 1/4 were wrinkled. Again, there was a 3-to-1 ratio. Remember 3-to-1. I'm going to probably, it's something you'll be seeing again and again in terms of these hybrid crosses. So an individual who has both a dominant and recessive allele, we call them a hybrid. So the F1 were hybrid. When two hybrids cross, they always end up with the offspring having the dominant to recessive ratio in a 3-to-1 ratio. So that was sort of the significance of Mendel's work. He disproved blending theory. He was one of the very first to mathematically analyze scientific data. I know you guys sometimes love and hate the relationship math has with what you do, but you can see how this mathematical relationship helped him to understand the mechanisms of inheritance. Again, Mendel published his paper. It wasn't very widely understood in his lifetime. It was published in about the 1860s. It wasn't until the 1900s till they recognized how really important what he really did was. So he came up with a couple principles, and he did repeat his cross experiments for all of the 7 pairs of traits. He always observed 100% of the F1s showed the dominant trait, and there was a 3-to-1 dominant to recessive ratio in the F2s. And so he used that to understand some basic principles, and these principles are something that we'll go over in some detail. The law of dominance, law of segregation. The law of dominance kind of tells you what it is. The law of dominance says that one characteristic can mask the presence of another. The law of segregation says then that when gametes are made, there's an equal chance of one or the other gametes being passed on to an offspring. So let's start off with Mendel's law of segregation. Again, that 3-to-1 indicated to him that each organism contains 2 factors for every trait. Those factors we now call alleles. So you have 2 factors for each trait. 2 true breeders have 2 identical factors. We say they're homozygous. And hybrids have 2 different traits. They're heterozygous. The 2 factors segregate during gamete formation. Each gamete receives only 1 factor. That makes sense in terms of what we understand with meiosis, so that those factors could be carried on chromosomes. And so if you're only going to have 1 factor, it makes sense that's what gametes are only haploid. The factors unite at fertilization, so each offspring has 2 factors. Again, this picture here looks an awful lot like some of the pictures that we talked about when we were talking about meiosis. Again, you have a diploid parent. In this case, the diploid parents are both, and when you look at the homologous pairs of chromosomes, the homologous pairs of chromosomes getting ready to divide carry the same allele or same factor. When the gametes from those 2 different lines meet, you have individuals who are hybrid or heterozygous carrying 2 different factors. So again, his law of dominance said the dominant factor is always expressed. But if it's always expressed, what that means is there's a difference between what an organism looks like and what the genes that they carry. So a dominant organism can be either heterozygous or homozygous. So when we talk about those individuals, we can describe their genetic makeup. When we're talking about the genetic makeup of an organism, its genotype. When we talk about what it looks like, we're talking about its phenotype. Again, hybrids and homozygous dominant true breeders look alike, and then the recessive phenotypes have to be homozygous for the recessive genotype. Now again, we've gone on beyond that because we're well past the 1850s. Scientists have experimentally confirmed all the conclusions that Mendel came to, but we now have a new vocabulary that we use to describe inheritance. Again, that idea of being diploid is well understood when we talk about meiosis. Diploid individuals have 2 alleles for each trait, and the traits we actually refer to as genes. So Mendel's factors are alleles, and they're carried on those homologous chromosomes, and then alleles sort during meiosis. Alleles can come in different types, the different forms of the same gene, and different alleles result from mutations. True breeders are homozygous, hybrids are heterozygous, and again, the gametes that are formed are haploid, or they don't have a homologous pair, they only have a single allele because of meiosis. So we can then look a little bit more clearly at inheritance. This is another one of Mendel's traits, those purple and white flowers. Here we have a true breeding purple and a true breeding white crossing, and we've now gone on to use something called a Punnett square to describe the trait. What we do is we figure out the possible gametes for each flower, and we put them on the top, in the case of the whites, or on the side of a chart, and then we fill in the chart by bringing 1 letter over and 1 letter down. When we're filling in Punnett squares, it's important that you always put, just like in a sentence, you always put the capital letter first. We always put the capital letter first because we know if there's a capital letter there, the capital letter refers to the dominant trait. And if I put the capital letter first, it lets you know that the individual is going to have the dominant phenotype Again, we're going to do quite a few Punnett squares over the course of the rest of the class periods. A test cross, real quick, is one of those things that we can do to determine if a dominant individual is homozygous or heterozygous. So if you have someone with a dominant trait, you cross them with someone with a recessive trait. The individual, the offspring that they have, cannot be recessive unless the individual with the dominant trait was heterozygous. And so that's referred to as a test cross, and here's a neat picture of a test cross. The next thing that Mendel went on to do is something called a dihybrid cross, where he looked at what happens when you look at two traits at the same time. In this case, he's looking at yellow and round seeds and crossing them with green and wrinkled seeds. And all the F1, again, just like previously, show the dominant form of each trait. They allowed the F1 to cross with themselves to self-fertilize and produce F2. And because we're talking about different alleles that are found on different chromosomes, they migrate separately, and there's many more possible combinations of offspring. So when he actually looked at what he got something that was a 9:3 to 3:1 ratio of the possible different types. Again, that had to do with the idea of independent assortment. Each of those factors or alleles sort independently from any other allele as long as they're found on different chromosomes. There's a way of figuring out possible gamete formation, and it's called the FOIL law. FOIL stands for first, outer, inner, and last. And again, when we go to do a dihybrid cross, you can use the FOIL method to figure out which alleles go on the outside of the Punnett square. Again, some more rules for doing Punnett squares. When you start to do a Punnett square, and this is a good sort of way to start any problem, you assign a letter for each trait. You write down the possible alternative traits. You assign a letter to each trait. Write it down. The recessive, you usually choose a letter that represents the recessive form of the trait. Again, one of the things that I always talk about is it's better to use a letter for something that has a different uppercase and lowercase appearance. Again, I'm happy with them choosing G's in this slide, but W is a little bit hard. They may have chosen something else. The letter R for wrinkled might have worked a little bit better, just because you could differentiate between uppercase and lowercase if you have sloppy handwriting. So this is a picture of what the dihybrid square looks like that comes up with, or the Punnett square looks like that comes up with from the dihybrid cross. And again, you can see the 9:3 to 3:1 ratio. We'll do, again, a couple of these in class, and we'll also do some for your homework. At possible crosses, we can look at flower color using purple and white as the different alleles. Again, we're using the letters W because white is the recessive allele. So which color is recessive? We've seen the purple and white after the first generation. Only purple appears. That means that white is recessive. Seeds can be round or wrinkled. Again, the big R we're using for seed shape. The reason we're using the big R for round, even though it's dominant, is we've already used the letter W, and so it makes sense to use that. So again, the big W stands for purple. And so if I ask you what the genotype of the first one is, you would expect that the genotype would be because it's got a big W, it's going to be purple, and because it's got a big R, it's going to be round. And so purple flowers, round seeds for the first one. The second one, again, you see the big W, that means you're going to have purple flowers, but because you have the two little R's, you end up with wrinkled seeds. In this one, white flowered, wrinkled seeds, white flowers and round seeds. We'll do lots of problems, let you play with this stuff, and you probably find that it really is pretty easy to figure out different genetics.